Hello and welcome to week 2 of the course on polymers. In this week, we are going to discuss properties of a single macromolecule. Uh, Before we get on to doing the discussion of the properties, we are looking at some sustainable aspects of polymeric systems, and we will look at in this lecture at polymerization and depolymerization in the context of sustainability. And this lecture is again also going to be more about thought process and the challenges that are with us and possibilities that can be exploited. So the focus will be on how do we think in terms of new ways of polymerization and depolymerization. Just to begin with, let's look at this exam question. In fact, very recent this year's exam question, where we have to look at a polymer and its corresponding polymerization. So, of course, for each polymer, we have a preferred polymerization method. And this is because it gives us control over all the aspects that we discussed in earlier lectures in terms of molar mass distribution, in terms of molecular architecture, in terms of sequencing. So whatever are the features needed in an eventual macromolecule, if they can be achieved using a given polymerization method, then it becomes preferred for that particular polymer. So in this particular question, for example, you can think in terms of the possibilities that are mentioned are coordination polymerization. I hope you can recall what is meant by coordination polymerization. What are examples of coordination polymerization? There are two examples of ionic polymerization, cationic and anionic. And then, of course, we spent in the sixth lecture time thinking about step growth polymerization. We looked at an example of polyester where an acid and alcohol reacted. So among these polymers which are mentioned, which one might have these reaction where there is a condensation product involved? So I'm sure you can rule out polyethylene, and maybe that's why, for example, it's right next to step growth. And so Q2 is definitely not an option. And so what are the possibilities that you can think of? So ponder over this, while the other thing that we've of course classified in the polymerization processes, we said that step growth is an important class of polymerization by which many polymers are made. The other broad class we looked at was in terms of activated center, where monomer by itself doesn't react, but in combination with an initiator or a catalyst, it does. So the example of activated species could be free radical or ions, or it could be catalytic. And we have already seen that Ziegler-Natta is very important in terms of making polyethylenes and polypropylene. And so it's an important discovery in terms of having good control on polymerization. In the last 20 years or so, we also have metallocene as an important class of materials which are become important for manipulating and having narrow molar mass distribution and controlling branching and so on. And you can see the highlight here that I want you to pick up is in terms of stereochemistry, in terms of what is the arrangement. Given a certain set of interactions that are possible with the three-dimensional structure of this catalyst, then monomers can come only in a particular way and get attached. And therefore, using these catalysts, whether coordination catalysts or metallocene catalysts, stereochemistry is managed in such a way that you get very good control on whatever is the polymerization product that is available. So you get control on the whether branches form or not, whether which type of tacticity is obtained, and so on. So we'll just look at ionic polymerization before we start looking at new ways of thinking about polymerization. The advantage of ionic polymerization is that the dispersity, which is the ratio of weight average molar mass to number average molar mass, can be close to 1, 1.05, 1.07, 1.1. Such polydispersity indices are possible when we use ionic polymerization. And this is commercially exploited also. So two most important examples of ionic polymerization are in terms of rubbers. So we use for styrene systems anionic polymerization, and it's used for styrene butadiene rubber. It is also used for another important copolymer of styrene, which is SBS. So there will be minor differences in terms of how the sequencing of monomers is. The monomers here are styrene and butadiene. So in this case, the active center is through an anion which comes from butyl lithium, and this class of polymerization is also called living polymerization. And in fact, that's the reason why control on molecular weight is so common and dispersity is obtained to be low. What happens in this case is monomer comes and attaches to the active center, and the active center will live as long as monomer is alive. The reaction stops in the end. Active center is still alive, but there is no monomer to react. And also, there are no side reactions which can cause the polymer to become a dead end. So, for example, two chains combine, which is called a termination step. That is not possible here. Or chain transfer. A growing chain suddenly becomes transfers some of its chain mass to another growing active center. So such chain transfer reactions are also not possible. So therefore, given that these reactions which are leading to molar mass distribution being wider are not there in this case, and secondly, all the monomer can get attached to active centers, we get very good control on molar mass distribution. The other common example of ionic polymerization, in this case cationic polymerization through active center of aluminum chloride, is butyl rubber. And in this case, though it's a butyl rubber based on isobutylene, for commercial systems small amounts of isoprene is added. Of course, the answer is there on the slide, but before you look at it, think why isoprene is added, given that we already discussed about isoprene being part of natural rubber. And of course, it is because you can cross-link it with sulfur. So in this case, molar mass is controlled by temperature, and depending on temperature, we get molar mass very high or low. And in this case also, termination reactions are not feasible, but chain transfer reactions can take place, and therefore molar mass distribution is not going to be as narrow as in case of anionic polymerization. Just to contrast the kind of dispersity that we get in ionic polymerization, this is the dispersity that you can obtain in step growth. And if you remember, P is the extent of reaction, is the number of functional groups that have reacted to the total number of functional groups that were present initially. So if you have a 99% reaction, D is going to be 1.99. So you can see that dispersity in this case is going to be close to 2 when the reaction goes to completion. As P approaches 1, reaction goes to completion. So therefore, D can be 2 when the reaction is complete. So therefore, the dispersity in step growth polymerization is always going to be closer to 2, while in ionic polymerization you can get very close to 1. And so this is one of the advantages of an ionic polymerization. So this question here from the exam is asking us to calculate the number average molar mass. And so given that this is the example of PET synthesis, this is based on a very simple theory of Carothers, where we can find out quickly what is the molar mass as a function of extent of polymerization. So wanted to pause here and try to solve this problem before you go further, but this is just to contrast the difference between what is step growth and ionic polymerization. So let's look at new ways of thinking about polymerization. Just pause and think, what is the current way? How do we do this now? So we currently we use fossil fuel, and we use several synthetic processes to get monomers, and these monomers are polymerized to get polymerization, and several plastics and rubbers and FRP are made. And once the use is over, we end up with a lot of waste. So this is the current paradigm that we think is what has led to so many questions being raised about plastics as sustainable material. So now can we think of a way in which this looks like a cyclic process? And so this is based on the reference that is cited here. At the end of the lecture, I'll also give you the complete reference. If you're interested, you can go and look further. So we have plant and biomass as a renewable resource, as we discussed in the last class already. So can we not keep that and keep a cyclic process in which from which we first get some small molecules. We could also use these to make monomers. Then once monomers are there, we could either use in vitro polymerization, which means in a lab with enzymes and other catalyst processes, but not in a bacteria or any other biological resource. And we can use chemical synthesis to get the polymers. And then the other possibility is we can get small molecules, and we can also do bacterial synthesis, the way we discussed in the previous lecture on renewable resources, and again get polymers. And once the use is done, post use, we can again very importantly think in terms of depolymerization using catalysts such as enzymes, depolymerize, or microorganisms. And then what we end up with is smaller fragments of molecules. So from using these two, we can again get small molecules, or we can eventually lead to biodegradation to carbon dioxide and water. And of course, this can again be reused. The carbon dioxide can be again photosynthesized by and to get plant and biomass. Some of the small molecules can again be reused. So you can see that now this is a cyclic process where from raw materials to post use, we have closed the cycle. So can we think of many of the polymeric systems that we are using can belong to such a cyclic process? And that's the challenge that we have in terms of sustainable polymers. So just looking at what are the challenges when we think of polymerization processes, for biopolymers and natural polymers, these are the examples of what are the strategies available in terms of control of molecular structure. So of course, molar mass itself is controlled. And in nature, we have examples of oligomeric unit, which means small number of repeating units, all the way to extremely high molar mass chains. So therefore, synthesis can be done to make a very small number of repeating units to extremely large number of repeating units. And also there is very nice control on molar mass distributions. And in fact, we get uniform or monodispersed polymers for various biopolymers. On the other hand, we also have examples of polysaccharides where broad molar mass distribution is also feasible. We will see that some of the applications, a broader molar mass distribution is desirable. As I've mentioned this earlier also, smaller molar mass present in a sample usually is good for processability from point of view of managing the rheological properties and viscosity in melt conditions. So therefore, it is from properties point of view, narrow molar mass distribution may be always desirable. But from processing point of view, sometimes smaller molar mass weight fraction may also be important. So as far as proteins and polynucleic acids are concerned, the control required is in terms of sequence. We have a set of monomers which are amino acids and nucleotides, and they have to be stitched together in a particular sequence. When it comes to polysaccharides, not only do we have sequence, we also have variety of features in terms of geometric linkages. So there is a very good diversity, and this is something which we will look at later in the course. There is a diversity of linkages which are possible. If you look at cellulose, starch, pectin, all of these are polysaccharides, but they have very different properties. And that is because of the diversity of monomers as well as diversity of linkages which are present in these kind of polysaccharides. And with polysaccharides, we also have possibility of branching and networking and cross-linking. So therefore, in biopolymer world itself, all the features that we have thought of in terms of synthetic molecular architecture is present. Now, can this be exploited in terms of polymerization techniques for making new set of polymers? So that's the question that is broadly in front of us. And so to address that, generally we can think of these two sets of techniques. In vivo polymerization, in which case we use bacterial polymerization, or in vitro polymerization, in which case we can use some enzymes as the catalyst that can be used for polymerization. And in case of bacterial polymerization, we have polymers produced within the cell also and produced in the cell, but then they are excreted out of the cell, so extracellular polymers. And there are four families of polymers which are made by bacterial cells. So polysaccharides is something which we have discussed quite a bit already in this course, polyesters as well. Polyhydroxybutyrate, the example that we saw in the last lecture, is an example of a bacterial polymer which is made by using in vivo polymerization. Bacteria can also make polyamides and polyphosphates. So these broad classes of polymers that are made. Of course, remember the challenge is we are talking about synthetic polymers, which are families of polyolefins, which are families of polyimides, which are families of polyether. So we are talking about families which are much larger set of families compared to what these are. Now the question is, can these families of polymers produce the set of properties that are already known to us? For example, Teflon as a very good engineering material, can any of the polymers which is made using such techniques can become and have properties like Teflon? And right now, of course, the answer is no. And that's where the challenge lies. So the other thing which is makes this a cyclic process, many of these polymers that are made using these strategies, is the fact that depolymerization is possible. And depolymerization is possible because of these enzymes, which attack the macromolecule and lead to chain scission or breakage of macromolecules. And once the smaller fragments of degradation products are available, these will be lower molar mass and smaller fragments. And so these can be then used by microbial cell as carbon and energy source. Just the way glucose or other sugar sources are used by cells, they can use these other smaller fragments which are coming from degradation of macromolecules. And once this happens, then eventually we have carbon dioxide and water. So from a macromolecule, we can go to carbon dioxide and water. So therefore, in vivo and in vitro polymerization has the capacity to give us macromolecules of a limited set of families presently. But the advantage is that you can close the cycle. Now, can this be done for larger set of polymers? And can we get engineering applications out of such polymers? That's a question to think of. The other challenging aspect that leads us to research and possibilities is in terms of controlled polymerization and depolymerization. So on demand, if we can polymerize something or depolymerize something, that is something like natural body. And another important example from body is self-healing. When we get a cut or a wound, it heals itself. So is it not possible in engineering domain that if we get crack on the material, can the crack not heal itself? And so if we have this ability of on-demand polymerization, it may be possible to develop what are called self-healing materials. And there are some examples of demonstrations, but this is a very important area of research in terms of trying to see if we can get self-healing polymers in terms of engineering materials, which are very promising to use because of the ability of not leading to catastrophic failure. But due to self-healing, they can remain functional and eventually we can replace it before a failure or an accident can happen. So with this thought, we can just close this lecture in terms of the questions to the answers that were posed. I hope all of you recognize that Ziegler-Natta is an example of coordination polymerization, and it is used for polyolefins. SBR is common example of anionic polymer, while cationic polymer is butyl rubber. And polycarbonate is produced by step growth polymerization using bisphenol A. So one of the things that you can think of is, do I have to memorize all of these things? And my answer would be no. You have, in terms of develop judgment and skill in terms of recognizing the patterns. So in case of polyolefins, you have to remember that we think in terms of control of branching, control of isotactic or tacticity. So we're talking about some geometric feature, and therefore a coordination catalyst. That's why it's called coordination. So there is some pattern in the way nomenclature is and what. So you have to try to remember the underlying ideas of polymerization synthesis, and it will become natural in terms of what the polymerization technique is. And of course, in terms of step growth polymerization, this is a simple formula to calculate the molar mass, average molar mass for a step growth polymerization. So with this, we will come to a close on this lecture, which is related to thinking about sustainability of polymeric systems. Thank you.